Chapter 1: Foundations of Animal Physiology. Animal physiology represents the comprehensive study of how living organisms function at the molecular, cellular, tissue, organ, and whole organism levels, examining the intricate mechanisms that enable animals to maintain life, respond to environmental challenges, and successfully reproduce. This discipline bridges the gap between the structural complexity revealed by anatomy and the dynamic processes that characterize living systems, providing fundamental insights into how biological machines operate under diverse conditions. Understanding animal physiology requires appreciation of both the universal principles that govern all life and the remarkable diversity of solutions that different species have evolved to address common physiological challenges. The concept of homeostasis stands as one of the central organizing principles of animal physiology, describing the dynamic equilibrium through which organisms maintain internal conditions within narrow ranges despite constant fluctuations in external environments. This regulatory capacity involves sophisticated feedback systems that detect deviations from optimal conditions and initiate corrective responses that restore balance. Claude Bernard first articulated the importance of internal constancy for life, noting that the stability of the internal environment provides the foundation for free and independent existence in variable external conditions. Negative feedback loops represent the primary mechanism through which homeostatic regulation operates, creating self-correcting systems that counteract disturbances and maintain stability. When body temperature rises above optimal levels, thermoreceptors detect the change and trigger responses, including vasodilation, sweating, and behavioral adjustments that promote heat loss and restore normal temperature. The effectiveness of these regulatory systems demonstrates the remarkable precision with which physiological processes can be controlled through coordinated cellular and molecular mechanisms. Positive feedback systems, while less common than negative feedback, play crucial roles in specific physiological processes where amplification rather than stability is required. During childbirth, the stretching of the cervix stimulates oxytocin release, which intensifies uterine contractions, leading to further cervical stretching and additional oxytocin release until delivery occurs. These self-reinforcing cycles demonstrate how physiological systems can be designed to achieve specific outcomes rather than maintain equilibrium. The hierarchical organization of physiological systems reflects the nested levels at which biological processes operate, from molecular interactions within individual cells to coordinated responses involving multiple organ systems. Emergent properties arise at each level of organization, creating capabilities that cannot be predicted from knowledge of component parts alone. The contractile properties of muscle tissue, for example, emerge from the coordinated interactions of contractile proteins, but cannot be understood solely from the properties of individual protein molecules. Comparative physiology examines how different species have solved similar physiological challenges through diverse evolutionary pathways, revealing both conserved mechanisms that reflect common ancestry and convergent solutions that demonstrate the constraints imposed by physical and chemical laws. The evolution of flight in birds, bats, and insects required similar solutions to problems of lift generation and metabolic fuel supply, yet each group achieved these capabilities through different anatomical and physiological modifications. Environmental physiology investigates how animals adapt to diverse physical and chemical conditions, including temperature extremes, oxygen availability, water balance, and dietary constraints. These adaptations often involve trade-offs between different physiological capabilities, as organisms optimize their performance for specific environmental conditions while accepting limitations in other areas. Arctic mammals maintain body temperature through enhanced insulation and metabolic heat production, but these adaptations may limit their heat dissipation capacity in warmer environments. The scaling of physiological processes with body size reveals fundamental relationships between organism size and biological function that constrain evolutionary possibilities while creating predictable patterns across diverse taxa. Surface area to volume ratios determine heat and gas exchange rates, while the scaling of metabolic rate with body mass affects energy requirements and life history patterns. These scaling relationships help explain why different-sized animals often require different physiological solutions to common problems. Physiological ecology examines how environmental factors influence physiological performance and how physiological constraints shape ecological relationships, including habitat selection, seasonal migrations, and species interactions. The physiological tolerance limits of different species often determine their geographic distributions and responses to environmental change, making physiological understanding crucial for predicting ecological responses to climate change and habitat modification. Molecular physiology investigates the cellular and molecular mechanisms that underlie physiological processes, revealing how genetic information is translated into functional capabilities and how environmental signals regulate gene expression and protein function. The development of molecular techniques has revolutionized physiological research by enabling investigation of mechanisms at previously inaccessible scales, while revealing the remarkable conservation of fundamental processes across diverse organisms. Systems physiology examines how different organ systems integrate their functions to maintain whole organism performance, recognizing that physiological processes rarely operate in isolation but rather function as components of complex networks involving multiple feedback loops and regulatory pathways. The coordination between cardiovascular, respiratory, and metabolic systems during exercise exemplifies how multiple systems must adjust their outputs to meet changing physiological demands. Experimental approaches in animal physiology range from non-invasive field measurements of free-living animals to detailed cellular and molecular studies of isolated tissues and organs. Each approach provides different types of information while involving specific limitations and assumptions that must be carefully considered when interpreting results. Laboratory studies offer precise control over experimental conditions, but may not reflect the complexity of natural environments, while field studies provide ecological relevance but often involve reduced experimental control. The application of engineering principles to physiological systems has provided powerful analytical tools for understanding how biological systems achieve their remarkable performance capabilities. Biomechanical analysis reveals how musculoskeletal systems generate force and movement, while fluid dynamic principles explain how cardiovascular and respiratory systems achieve efficient transport of gases and nutrients. These engineering perspectives often reveal optimization principles that help explain evolutionary patterns in physiological design. Evolutionary physiology examines how physiological traits have evolved in response to environmental pressures and how physiological constraints have influenced evolutionary trajectories. This perspective recognizes that current physiological capabilities reflect both adaptive responses to environmental challenges and historical constraints imposed by evolutionary ancestry. Understanding these evolutionary influences is crucial for predicting how species might respond to novel environmental challenges. The medical relevance of animal physiology extends beyond basic scientific understanding to practical applications in human and veterinary medicine, conservation biology, and biotechnology. Comparative physiological studies have provided crucial insights into human disease mechanisms while revealing potential therapeutic targets and treatment strategies. Understanding physiological diversity also informs conservation efforts by revealing the specific environmental requirements needed to maintain viable populations of threatened species. Chapter 2: Circulatory and Respiratory Systems. The circulatory and respiratory systems work in intimate coordination to deliver oxygen and nutrients to every cell in the animal body, while removing metabolic wastes and carbon dioxide, creating the internal transport networks that enable complex multicellular life. These systems demonstrate remarkable evolutionary diversity in their anatomical organization and functional capabilities, reflecting the different solutions that various animal groups have developed to address the fundamental challenge of maintaining adequate gas exchange and nutrient delivery in organisms ranging from tiny invertebrates to massive vertebrates. Understanding these systems reveals both the constraints imposed by physical laws and the ingenious biological innovations that have overcome apparent limitations. Cardiovascular systems encompass the heart, blood vessels, and blood that together create pressurized circulation, capable of delivering materials throughout the body with precise temporal and spatial control. The evolution of efficient circulatory systems enabled the development of large, metabolically active organisms by solving the diffusion limitations that constrain the size of simpler animals. Single-celled organisms can rely on diffusion for gas and nutrient exchange, but multicellular animals require active transport mechanisms to service cells located far from exchange surfaces. Heart structure and function vary dramatically across animal taxa, reflecting different evolutionary solutions to the challenge of generating adequate circulation pressure while maintaining efficiency and reliability. Fish hearts contain two chambers that create a single circulation loop, pumping deoxygenated blood through the gills where gas exchange occurs before distributing oxygenated blood throughout the body. This arrangement creates relatively low systemic blood pressure, but proves adequate for the metabolic demands of ectothermic aquatic life. Mammalian and bird hearts evolved four-chambered designs that separate pulmonary and systemic circulation, enabling higher blood pressures and more efficient oxygen delivery that supports the elevated metabolic rates required for endothermic temperature regulation. The complete separation of oxygenated and deoxygenated bloodstreams maximizes oxygen delivery efficiency, while allowing independent regulation of pulmonary and systemic blood flow to match varying physiological demands. Reptilian hearts demonstrate intermediate designs with functional separation of oxygenated and deoxygenated blood, despite incomplete anatomical separation, revealing evolutionary transitions between different circulatory architectures. Some reptiles can selectively bypass pulmonary circulation during diving or other periods when gas exchange is limited, demonstrating the physiological flexibility that can be achieved through modified circulatory designs. Blood composition and properties reflect the diverse functions that this remarkable tissue performs, including oxygen transport, immune defense, nutrient distribution, waste removal, and chemical communication between distant body regions. Hemoglobin and other respiratory pigments have evolved multiple times to enhance oxygen carrying capacity, with different molecular structures providing optimal performance under specific environmental conditions, including high altitude, cold temperatures, and variable oxygen availability. Oxygen binding curves for different hemoglobins reveal sophisticated molecular adaptations that optimize oxygen transport under specific physiological conditions. Fetal hemoglobin displays higher oxygen affinity than adult hemoglobin, facilitating oxygen transfer across the placenta from maternal to fetal circulation. High-altitude animals often possess hemoglobins with increased oxygen affinity that maintain adequate oxygen uptake despite reduced atmospheric pressure. Vascular architecture demonstrates remarkable engineering sophistication in its hierarchical branching patterns that minimize energy expenditure while maximizing transport efficiency. The systematic reduction in vessel diameter from arteries through arterioles to capillaries creates optimal conditions for nutrient and gas exchange while maintaining adequate flow rates throughout the circulation network. Capillary density varies among different tissues in proportion to their metabolic demands, with highly active tissues receiving more extensive vascular networks. Blood pressure regulation involves complex integrative mechanisms that coordinate cardiac output, vascular resistance, and blood volume to maintain adequate perfusion of vital organs while accommodating varying physiological demands. Baroreceptors in major arteries continuously monitor blood pressure and initiate reflex adjustments in heart rate, cardiac contractility, and vascular tone that compensate for pressure changes. These feedback systems demonstrate remarkable precision in maintaining circulatory homeostasis. Respiratory systems evolved diverse anatomical structures and functional strategies to maximize gas exchange efficiency, while minimizing energy costs and structural investments. The fundamental challenge involves creating large surface areas for diffusion, while maintaining thin barriers that separate internal and external environments. Different animal groups have achieved these requirements through gills, lungs, tracheae, and other specialized structures that reflect their evolutionary histories and environmental adaptations. Aquatic respiration through gills demonstrates elegant solutions to the challenge of extracting dissolved oxygen from water, which contains much lower oxygen concentrations than air while requiring more energy to move due to its higher density and viscosity. Fish gills achieve remarkable efficiency through countercurrent flow arrangements that maintain concentration gradients along the entire length of gas exchange surfaces, enabling oxygen extraction rates that approach theoretical maximums. Terrestrial respiration faces different challenges related to preventing water loss, while maintaining adequate gas exchange in environments where oxygen is abundant, but desiccation poses significant risks. Mammalian lungs create vast internal surface areas through branching airways that terminate in microscopic alveoli surrounded by dense capillary networks. This design maximizes gas exchange capacity, while minimizing water loss through physiological dead space and humidification mechanisms. Ventilation mechanisms range from simple diffusion and ciliary currents in small animals to sophisticated muscular pumping systems in larger organisms that actively move respiratory media across exchange surfaces. Mammalian breathing involves coordinated contractions of the diaphragm and intercostal muscles that create pressure changes driving air movement, while many fish use buccal and opercular pumps that force water across gill surfaces in unidirectional flow patterns. Respiratory control systems integrate information about blood gas concentrations, pH levels, and metabolic demands to regulate ventilation rates and patterns that maintain adequate gas exchange under varying conditions. Central chemoreceptors in the brainstem respond to carbon dioxide and pH changes, while peripheral chemoreceptors monitor oxygen levels, creating feedback loops that automatically adjust breathing to match metabolic requirements. High-altitude adaptations reveal the remarkable plasticity of respiratory and circulatory systems in responding to chronic hypoxic conditions. Individuals acclimated to high altitude develop increased lung capacity, enhanced ventilation rates, elevated red blood cell production, and improved oxygen extraction efficiency that partially compensates for reduced atmospheric oxygen availability. These adaptations demonstrate the integrated responses that maintain physiological performance under challenging environmental conditions. Diving physiology presents extreme examples of respiratory adaptation, involving breath-holding capabilities that far exceed normal mammalian limits. Marine mammals achieve extended dive durations through elevated blood and muscle oxygen stores, reduced metabolic rates, and selective blood flow redistribution that prioritizes vital organs while temporarily limiting circulation to less critical tissues. These adaptations enable exploitation of aquatic resources while maintaining air-breathing respiratory systems. Exercise physiology reveals the remarkable capacity of circulatory and respiratory systems to increase their output in response to elevated metabolic demands. During intense exercise, cardiac output can increase several-fold through elevated heart rate and stroke volume, while ventilation rates increase dramatically to maintain adequate gas exchange. The coordinated responses of these systems enable sustained high-intensity activity that would be impossible without precise physiological integration. Pathophysiology of circulatory and respiratory disorders provides insights into normal system function, while highlighting the clinical importance of understanding these physiological processes. Heart disease, hypertension, asthma, and other conditions result from disruptions in normal regulatory mechanisms, emphasizing how health depends on the proper functioning of these integrated systems. Chapter 3: Nervous System and Sensory Biology. The nervous system represents the most complex and sophisticated control network in animal biology, integrating sensory information from internal and external environments, while coordinating responses that enable survival, reproduction, and adaptation to changing conditions. This remarkable system evolved from simple nerve nets in primitive animals to the elaborate centralized nervous systems found in vertebrates, creating information processing capabilities that range from basic reflexes to complex cognitive functions, including learning, memory, and consciousness. Understanding nervous system function requires examination of both the fundamental properties of individual neurons and the emergent capabilities that arise from neural network interactions. Neurons serve as the fundamental functional units of nervous systems, specialized cells that have evolved unique capabilities for rapid electrical and chemical communication across long distances. The basic neuronal architecture includes dendrites that receive signals from other neurons, a cell body containing the nucleus and most cellular organelles, and an axon that transmits signals to target cells through specialized synaptic connections. This polarized structure enables directional information flow while allowing for complex integration of multiple input signals. Action potentials represent the universal language of nervous systems, providing the electrical signals that enable rapid communication between neurons and their target cells. These brief electrical events result from coordinated opening and closing of voltage-gated ion channels that create transient changes in membrane permeability and electrical potential. The all-or-nothing nature of action potentials ensures reliable signal transmission over long distances, while frequency coding allows neurons to convey information about stimulus intensity and duration. Synaptic transmission involves the conversion of electrical signals into chemical messages that can cross the gaps between neurons, enabling communication within neural networks while providing opportunities for signal modification and integration. The release of neurotransmitter molecules from presynaptic terminals creates chemical signals that bind to receptors on postsynaptic cells, producing either excitatory or inhibitory effects that influence the likelihood of action potential generation in target neurons. Neurotransmitter diversity reflects the sophisticated chemical communication systems that have evolved to provide specific and nuanced control over neural function. Different neurotransmitters produce distinct effects on target cells, while operating through various receptor mechanisms that can amplify, diminish, or modify synaptic signals. The same neurotransmitter can produce different effects depending on the specific receptors and signaling pathways present in target cells, creating tremendous flexibility in neural communication. Neural development involves remarkable processes through which the nervous system constructs itself, from relatively simple beginnings to create the precise connectivity patterns required for proper function. During embryonic development, neurons migrate to appropriate locations, extend axons that must navigate complex pathways to reach their targets, and form specific synaptic connections that wire the nervous system for its mature functions. These developmental processes involve molecular guidance mechanisms and activity-dependent refinement that sculpt neural circuits. Neuroplasticity enables nervous systems to modify their structure and function in response to experience, providing the biological foundation for learning, memory, and adaptation to environmental changes. Synaptic plasticity involves changes in the strength of connections between neurons based on their activity patterns, while structural plasticity can involve the formation of new synapses or even the generation of new neurons in some brain regions throughout life. Central nervous system organization varies dramatically across animal taxa, while reflecting common principles of functional specialization and hierarchical control. Simple nervous systems like those found in cnidarians consist of nerve nets that provide local coordination without centralized control, while more complex systems show increasing centralization and regionalization that enables more sophisticated information processing and behavioral control. Brain evolution demonstrates progressive increases in size and complexity that correlate with enhanced behavioral capabilities and environmental adaptability. The vertebrate brain shows characteristic regional specialization, including the hindbrain that controls basic life functions, the midbrain that integrates sensory and motor information, and the forebrain that reaches its greatest development in mammals where it supports complex cognitive functions including learning, memory, and decision-making. Sensory systems provide the crucial interface between animals and their environments, converting physical and chemical stimuli into neural signals that can be processed by the nervous system to guide behavior and physiological responses. The diversity of sensory capabilities across animal species reflects both the variety of environmental information that can be biologically useful and the remarkable evolutionary innovations that have enabled detection of signals ranging from single photons to magnetic fields. Vision represents perhaps the most thoroughly studied sensory modality, revealing fundamental principles of sensory transduction while demonstrating the sophisticated processing capabilities of neural systems. Photoreceptor cells contain specialized proteins that undergo conformational changes when exposed to light, initiating cascades of biochemical events that ultimately generate electrical signals proportional to light intensity. The evolution of different photoreceptor types enables color vision and adaptation to different light environments. Compound eyes found in arthropods demonstrate alternative solutions to the challenges of visual system design, creating wide fields of view and exceptional motion detection capabilities through arrays of individual optical units called ommatidia. While compound eyes typically provide lower spatial resolution than vertebrate camera eyes, they excel at detecting movement and can sample larger portions of the visual environment simultaneously. Auditory systems convert sound waves into neural signals through sophisticated mechanical transduction mechanisms that can detect incredibly small vibrations while discriminating between different frequencies and intensities. Mammalian hearing involves the coordinated function of outer, middle, and inner ear structures that amplify sound waves, convert them to mechanical vibrations, and ultimately stimulate hair cells that generate neural signals encoding acoustic information. Echolocation represents one of the most remarkable sensory adaptations, enabling animals like bats and dolphins to navigate and hunt using self-produced sounds and their reflected echoes. These biological sonar systems achieve spatial resolution that rivals or exceeds human-engineered systems, while operating in real time during rapid locomotion and complex behaviors. The neural processing required for echolocation reveals extraordinary computational capabilities. Chemical senses, including taste and smell, provide crucial information about food quality, predator presence, potential mates, and other chemically mediated aspects of the environment. Chemoreceptor cells contain specialized proteins that bind specific molecular structures, creating highly selective detection systems that can discriminate between countless different chemical compounds while maintaining sensitivity to very low concentrations. Mechanosensation encompasses diverse forms of touch, pressure, vibration, and proprioceptive sensing that provide information about physical interactions with the environment and the position and movement of body parts. Mechanoreceptors contain specialized structures that convert mechanical forces into electrical signals, enabling detection of stimuli ranging from gentle touches to potentially damaging pressures. Specialized senses found in various animal groups demonstrate the remarkable evolutionary innovations that have expanded sensory capabilities beyond the traditional 5 senses familiar to humans. Electroreception enables some fish to detect electrical fields generated by other organisms, while magnetoreception may involve specialized cells containing magnetic particles that function as biological compasses for navigation. Sensory integration involves complex neural processing that combines information from multiple sensory modalities to create coherent perceptions of environmental conditions, while filtering out irrelevant information and enhancing detection of biologically significant stimuli. The brain constructs perceptual representations that may differ significantly from the actual physical properties of stimuli, revealing the active role of neural processing in shaping sensory experience. Pain perception serves crucial protective functions by motivating avoidance of potentially damaging stimuli, while creating aversive experiences that promote learning about environmental hazards. Pain involves specialized sensory receptors that detect tissue damage or potentially harmful stimuli, but the subjective experience of pain emerges from complex neural processing that can be influenced by psychological and social factors. Sensory disorders and their treatment provide insights into normal sensory function, while highlighting the medical and social importance of understanding these biological systems. Hearing loss, blindness, chronic pain, and other sensory impairments significantly impact quality of life, while while research into sensory prosthetics and restoration techniques offers hope for therapeutic interventions based on detailed understanding of sensory system function. Chapter 4: Metabolism and Energy Balance. Metabolism encompasses the vast network of chemical reactions that occur within living organisms to maintain life, generate energy, and enable growth, reproduction, and adaptation to environmental challenges. These intricate biochemical processes involve the coordinated breakdown of nutrients to release stored energy and the synthesis of complex molecules needed for cellular structure and function. Understanding metabolism requires appreciation of both the universal biochemical pathways that characterize all life and the remarkable diversity of metabolic strategies that different organisms have evolved to thrive in diverse environments ranging from deep ocean vents to Arctic tundra. Energy metabolism forms the foundation of all biological processes, involving the conversion of chemical energy stored in food molecules into forms that can power cellular work, including biosynthesis, transport, movement, and information processing. The fundamental currency of cellular energy involves adenosine triphosphate, ATP, and other high-energy phosphate compounds that store and transfer energy through phosphorylation and dephosphorylation reactions. This energy storage system enables cells to capture energy from nutrient breakdown and deploy it when and where needed for cellular functions. Cellular respiration represents the primary pathway through which animals extract energy from nutrients, involving the systematic breakdown of glucose and other organic molecules through glycolysis, the citric acid cycle, and oxidative phosphorylation. This process demonstrates remarkable efficiency in energy extraction while generating carbon dioxide and water as end products. The evolution of aerobic respiration enabled organisms to extract far more energy from nutrients than anaerobic processes, supporting the evolution of complex multicellular life. Metabolic rate reflects the overall energy expenditure of organisms and varies enormously both between species and within individuals, depending on body size, activity level, environmental conditions, and physiological state. Basal or resting metabolic rate represents the minimum energy required to maintain basic physiological functions, including circulation, respiration, cellular maintenance, and temperature regulation in endothermic animals. Understanding metabolic rate variations provides insights into energy budgets and life history strategies. Scaling relationships reveal fundamental principles governing the relationship between body size and metabolic rate, with larger animals typically having higher absolute metabolic rates but lower mass-specific metabolic rates than smaller animals. These scaling laws reflect biophysical constraints, including surface area to volume ratios, diffusion limitations, and mechanical factors that influence how biological systems can be scaled up or down while maintaining functionality. Temperature effects on metabolism demonstrate the profound influence of environmental conditions on physiological processes, with most biochemical reactions roughly doubling in rate for every 10 degrees Celsius temperature increase. Ectothermic animals show dramatic metabolic responses to temperature changes, while endothermic animals maintain relatively stable body temperatures through metabolic heat production and behavioral thermoregulation. These different strategies represent alternative solutions to temperature challenges. Metabolic adaptation involves physiological adjustments that enable organisms to maintain energy balance under varying environmental conditions, including food scarcity, temperature extremes, and seasonal changes in resource availability. Some animals undergo dramatic metabolic suppression during hibernation or estivation, reducing energy expenditure to levels that can be sustained on stored body reserves for months. These adaptations demonstrate remarkable metabolic flexibility. Nutritional physiology examines how animals obtain, process, and utilize nutrients from their diets while addressing the challenges of extracting essential compounds from diverse food sources. Different feeding strategies, including herbivora, carnivora, and omnivora, require specialized digestive systems and metabolic pathways that reflect the nutritional composition and digestibility of different food types. The evolution of these specializations has enabled animals to exploit virtually every available energy source. Digestive system diversity reflects the remarkable range of solutions that animals have evolved for breaking down food and absorbing nutrients, from simple intracellular digestion in single-celled organisms to complex multi-chambered stomachs in ruminants that harbor symbiotic microorganisms capable of digesting cellulose. The length, structure, and enzymatic capabilities of digestive systems correlate with dietary habits and nutritional requirements. Enzyme systems involved in digestion and metabolism demonstrate sophisticated biochemical machinery that has evolved to process specific nutrients while operating efficiently under physiological conditions. Digestive enzymes including amylases, proteases, and lipases break down complex food molecules into simpler compounds that can be absorbed, while metabolic enzymes facilitate the countless reactions involved in energy production and biosynthesis. Nutrient absorption mechanisms involve specialized transport systems that move digested nutrients from the digestive tract into the bloodstream, where they can be distributed throughout the body. Different nutrients require different absorption mechanisms, from simple diffusion for some compounds to active transport systems that can concentrate nutrients against concentration gradients. The efficiency of these systems determines nutritional status and energy availability. Metabolic regulation involves complex control mechanisms that coordinate energy production and utilization while maintaining homeostasis despite varying energy demands and nutrient availability. Hormonal control systems, including insulin, glucagon, and others, provide systemic regulation of metabolism, while local regulatory mechanisms fine-tune metabolic activity in specific tissues and cellular compartments. Energy storage strategies enable animals to buffer periods of energy shortage by accumulating reserves during times of abundance, with different storage forms providing distinct advantages and limitations. Glycogen provides readily available energy storage but has limited capacity, while lipids offer high energy density storage that can sustain organisms through extended periods of food scarcity. Protein can serve as an energy source, but typically represents a last resort due to its structural importance. Thermal physiology involves the complex interactions between metabolism, behavior, and environmental temperature that determine how animals maintain optimal body temperatures for physiological function. Endothermic animals generate metabolic heat to maintain elevated body temperatures, while ectothermic animals rely primarily on behavioral thermoregulation and tolerance of temperature variation. Each strategy involves specific costs and benefits. Exercise physiology reveals how metabolic systems respond to increased energy demands during physical activity, involving coordinated adjustments in circulation, respiration, and cellular metabolism that enable sustained muscle contraction and movement. Different types of exercise place different demands on metabolic systems, from anaerobic power for brief intense efforts to aerobic endurance for sustained activities. Metabolic disorders provide insights into normal metabolic function, while highlighting the medical importance of understanding these physiological processes. Diabetes, obesity, metabolic syndrome, and other conditions result from disruptions in normal metabolic regulation, emphasizing how health depends on proper coordination of energy balance and nutrient utilization. Comparative metabolism reveals how different animal groups have evolved distinct metabolic strategies that reflect their evolutionary histories and ecological adaptations. Cold-adapted animals often have enhanced metabolic capacity for heat production, while desert animals may have reduced metabolic rates to minimize water and energy requirements. These comparisons illuminate the evolutionary flexibility of metabolic systems. Environmental metabolism examines how human activities and environmental changes affect animal metabolism through pollution, climate change, and habitat modification. Understanding these effects is crucial for predicting how animals will respond to environmental challenges, while developing conservation strategies that account for metabolic requirements and constraints. Chapter 5: Osmoregulation and Endocrine Control. Osmoregulation encompasses the sophisticated physiological mechanisms that enable animals to maintain optimal water and salt balance despite living in environments where water and solute concentrations vary dramatically from those required for proper cellular function. This fundamental aspect of animal physiology involves complex interactions between specialized organs, transport systems, and regulatory mechanisms that collectively ensure that internal fluid composition remains within narrow ranges compatible with life. The diversity of osmoregulatory strategies reflects the remarkable evolutionary solutions that different animal groups have developed to cope with environments ranging from marine waters with high salt concentrations to freshwater systems with minimal dissolved solutes to terrestrial environments where water conservation becomes paramount. Water balance represents one of the most critical physiological challenges facing all animals, as virtually every cellular process depends on appropriate hydration levels, while water loss or gain can rapidly disrupt cellular function and threaten survival. Animals must balance water intake from food, drink, and metabolic processes against water loss through excretion, respiration, and across body surfaces, requiring precise regulatory mechanisms that can adjust these processes to maintain homeostasis under varying environmental conditions. Osmotic regulation involves controlling the concentration of dissolved solutes in body fluids to prevent damaging water movements that would occur if cellular and extracellular fluid compositions were not properly balanced. Cells contain numerous proteins, nucleic acids, and other large molecules that cannot easily cross cell membranes, creating osmotic pressure that would draw water into cells and cause them to swell and burst if not counterbalanced by appropriate solute concentrations in extracellular fluids. Marine animals face unique osmoregulatory challenges due to the high salt concentration of seawater, which tends to draw water from their bodies while loading them with excess salts that can disrupt cellular function if allowed to accumulate. Different marine animal groups have evolved distinct strategies, including salt excretion through specialized glands, production of organic osmolytes that counterbalance inorganic salts, and behavioral adaptations that minimize salt exposure. Freshwater animals confront opposite challenges, as their environments contain very low salt concentrations that create osmotic gradients, favoring water influx and salt loss, requiring active mechanisms to pump out excess water while retaining essential salts. Many freshwater animals have evolved highly efficient kidneys, capable of producing large volumes of dilute urine, while reabsorbing valuable salts and nutrients. Terrestrial animals must cope with the constant threat of dehydration, while maintaining adequate hydration for physiological processes, leading to evolution of water conservation mechanisms, including efficient kidneys, reduced respiratory water loss, and behavioral strategies for obtaining water and avoiding excessive losses. Desert animals demonstrate extreme adaptations, including production of highly concentrated urine, dry feces, and minimal respiratory water loss. Kidney structure and function vary dramatically across animal taxa, while reflecting the osmoregulatory challenges faced by different groups, from simple filtration systems in invertebrates to complex multi-stage processing organs in vertebrates that can precisely regulate water and solute excretion. Mammalian kidneys achieve remarkable concentration capabilities through countercurrent multiplication systems that enable production of urine several times more concentrated than blood. Hormonal control of osmoregulation involves complex signaling systems that coordinate the activities of multiple organs, including kidneys, gills, salt glands, and digestive systems to maintain appropriate water and salt balance. Antidiuretic hormone, aldosterone, and other regulatory molecules provide systemic control over water and sodium handling, while enabling rapid adjustments to changing physiological demands and environmental conditions. The endocrine system represents a sophisticated communication network that uses chemical messengers called hormones to coordinate physiological processes across different organs and tissues, enabling integrated responses to environmental challenges and internal requirements. Unlike the rapid electrical signaling of the nervous system, endocrine communication typically involves slower but longer-lasting effects that are crucial for regulating growth, reproduction, metabolism, and homeostasis. Hormone chemistry encompasses diverse molecular structures, including proteins, peptides, steroids, and modified amino acids that provide chemical signals with varying properties, including solubility, stability, and receptor binding characteristics. These chemical differences determine how hormones are transported in the blood, how they interact with target cells, and how their effects are regulated and terminated. Endocrine glands represent specialized organs dedicated primarily to hormone production and secretion, while many other tissues produce hormones as secondary functions alongside their primary roles. The pituitary gland serves as a master regulator that controls many other endocrine organs, while glands including the thyroid, adrenals, and gonads produce hormones with specific regulatory functions. Hormone receptors determine which cells respond to particular hormones and what types of responses occur, with receptor distribution and sensitivity providing precise control over hormone effects. Some hormones bind to receptors on cell surfaces and activate second messenger systems, while others enter cells and directly influence gene expression through nuclear receptors. Feedback control systems regulate hormone production and release through negative feedback loops that maintain appropriate hormone levels, while preventing excessive responses that could disrupt homeostasis. The hypothalamic pituitary axis exemplifies sophisticated feedback control, with brain regions monitoring body conditions and adjusting hormone production accordingly. Growth hormone regulation demonstrates how endocrine systems coordinate complex developmental processes through multiple hormone interactions that promote tissue growth while ensuring appropriate body proportions and timing. Growth hormone stimulates production of growth factors while being regulated by releasing and inhibiting hormones from the hypothalamus. Stress responses involve coordinated endocrine and neural mechanisms that enable animals to cope with challenges, including predation, injury, disease, and environmental extremes. The activation of stress response systems mobilizes energy reserves, enhances cardiovascular function, and modifies behavior while temporarily suppressing non-essential functions including growth and reproduction. Reproductive endocrinology involves complex hormone cycles that coordinate gamete production, mating behaviors, pregnancy and parental care, while synchronizing reproductive activities with optimal environmental conditions. Sex hormones influence not only reproductive organs, but also secondary sexual characteristics, behavior patterns, and metabolic processes that support reproductive success. Thyroid hormones regulate metabolic rate, development, and numerous physiological processes through effects on gene expression and cellular metabolism. These hormones play crucial roles in temperature regulation, growth, and behavioral development, while demonstrating how endocrine signals can have widespread effects throughout the body. Calcium regulation involves precise hormonal control of calcium absorption, excretion, and mobilization from skeletal reserves to maintain blood calcium levels within narrow ranges required for proper muscle and nerve function. Parathyroid hormone, calcitonin, and vitamin D work together to regulate calcium homeostasis through effects on kidneys, intestines, and bone. Endocrine disruption by environmental pollutants has become an increasing concern, as numerous chemicals in the environment can interfere with normal hormone function, leading to reproductive problems, developmental abnormalities, and metabolic disorders. Understanding these effects requires knowledge of normal endocrine function and the mechanisms through which synthetic chemicals can disrupt hormonal signaling. Comparative endocrinology reveals both the conservation of fundamental hormonal mechanisms across diverse animal taxa and the evolutionary modifications that have adapted endocrine systems to specific ecological requirements. Many hormones show remarkable conservation of structure and function across vast evolutionary distances, while others have evolved specialized roles in particular animal groups. Chronobiology examines how endocrine systems regulate biological rhythms, including daily cycles of hormone release, that coordinate physiological processes with environmental light-dark cycles and seasonal changes. Circadian and circannual rhythms involve complex interactions between the nervous and endocrine systems that enable animals to anticipate and prepare for predictable environmental changes. Clinical applications of endocrine and osmoregulatory knowledge have led to numerous medical advances, including hormone replacement therapies, treatments for diabetes and other metabolic disorders, and management of kidney disease and fluid balance disorders. Understanding these physiological systems provides the foundation for diagnosing and treating numerous health conditions while developing new therapeutic approaches based on detailed knowledge of normal regulatory mechanisms. Marine Biology, the study of ocean life and ecosystems. By Ray Arders. Chapter 1. The Ocean Environment and its Inhabitants. The ocean represents Earth's largest habitat, covering approximately 71% of the planet's surface and containing over 99% of the biosphere's living space by volume. This vast aquatic realm encompasses environments ranging from sunlit surface waters to the perpetual darkness of abyssal depths, from tropical coral reefs to ice-covered polar seas, each supporting unique communities of organisms that have evolved remarkable adaptations to thrive in the marine environment. The physical and chemical properties of seawater create the fundamental conditions that shape marine life, with salinity, temperature, pressure, and light availability serving as primary factors influencing the distribution and evolution of marine organisms. Seawater's average salinity of 35 parts per thousand creates an osmotic challenge for most organisms, requiring specialized physiological mechanisms to maintain water balance and cellular function in this hyperosmotic environment. Temperature stratification in the ocean creates distinct thermal zones that profoundly influence marine ecosystems, with the thermocline serving as a barrier that separates warm surface waters from cold deep waters in most regions. This thermal structure affects nutrient distribution, oxygen levels, and organism distribution, creating vertical zonation patterns that are among the most fundamental organizing principles in marine ecology. Hydrostatic pressure increases dramatically with depth, rising by approximately 1 atmosphere every 10 meters, creating extreme pressure conditions that exceed 1,000 atmospheres in the deepest ocean trenches. Marine organisms have evolved sophisticated adaptations to function under these pressure regimes, including specialized proteins, modified gas-filled organs, and unique biochemical processes that remain functional under crushing pressure. Light penetration in seawater is limited by absorption and scattering, with different wavelengths being absorbed at different rates, creating distinct optical environments at various depths. The euphotic zone, where sufficient light exists for photosynthesis, typically extends only to depths of 100 to 200 meters, while the vast majority of the ocean exists in perpetual darkness where organisms must rely on chemical energy sources or descending organic matter. Ocean circulation patterns create global conveyor belts that transport heat, nutrients, and organisms across vast distances, connecting distant marine ecosystems and influencing climate patterns worldwide. Surface currents driven by wind patterns and deep water circulation, driven by density differences, create complex three-dimensional flow patterns that affect everything from nutrient distribution to larval dispersal. Marine primary productivity forms the foundation of ocean food webs through photosynthesis by microscopic phytoplankton that convert inorganic carbon and nutrients into organic matter using sunlight energy. These tiny organisms, including diatoms, dinoflagellates, and cyanobacteria, are responsible for approximately half of global photosynthesis and support virtually all marine food chains through their role as primary producers. The concept of marine biodiversity encompasses not only the spectacular megafauna that capture public attention, but also the vast diversity of microscopic organisms that constitute the majority of marine species. Current estimates suggest that the ocean may contain millions of species, with new discoveries continuing at a rapid pace as exploration technologies advance and previously inaccessible habitats are studied. Endemic species distributions in marine environments often reflect geological history, ocean circulation patterns, and evolutionary processes that have isolated populations and promoted speciation. Island biogeography principles apply to marine systems, with isolated seamounts, oceanic islands, and enclosed seas serving as laboratories for evolution and centers of endemism. Marine food webs exhibit complex structures that reflect the three-dimensional nature of the ocean environment and the diverse feeding strategies employed by marine organisms. Primary consumers include zooplankton that graze on phytoplankton, filter-feeding invertebrates that capture suspended particles, and herbivorous fish that consume marine algae, while higher trophic levels include predatory fish, marine mammals, and seabirds. Trophic cascades in marine ecosystems demonstrate how changes at one trophic level can propagate through food webs with sometimes unexpected consequences. The removal of top predators can lead to population explosions of their prey species, which may then overconsume their own food sources, ultimately affecting primary producers and ecosystem structure. Pelagic and benthic realms represent fundamentally different marine environments, with the water column supporting communities of swimming and floating organisms, while the seafloor harbors communities adapted to life on or in sediments. These realms are connected through the vertical migration of organisms and the settling of organic matter from surface waters to the deep sea. Hydrothermal vents and cold seeps represent chemosynthetic ecosystems that derive energy from chemical reactions rather than photosynthesis, supporting unique communities of organisms that thrive in conditions previously thought to be incompatible with life. These ecosystems have revolutionized our understanding of life's limits and may provide insights into early evolution and potential extraterrestrial life. Symbiotic relationships pervade marine ecosystems, from the zooxanthellae that provide energy to coral polyps through photosynthesis, to the bacterial symbionts that enable deep-sea organisms to exploit chemical energy sources. These partnerships often involve intimate physiological integration and have been crucial for the evolution of complex marine ecosystems. Marine conservation challenges include habitat destruction, overfishing, pollution, climate change, and ocean acidification, all of which threaten the integrity of marine ecosystems and the services they provide to human society. Understanding marine biology is essential for developing effective conservation strategies that can protect marine biodiversity while supporting sustainable human use of ocean resources. Chapter 2: Marine Plankton and Microscopic Life. Marine plankton comprise the vast community of organisms that drift with ocean currents, ranging from microscopic bacteria and protists to larger invertebrates and fish larvae that spend all or part of their lives as members of the planktonic community. These organisms form the foundation of marine food webs while playing crucial roles in global biogeochemical cycles, making them among the most important and abundant life forms on Earth. Phytoplankton represent the ocean's primary producers, converting inorganic carbon dioxide into organic compounds through photosynthesis and forming the base of virtually all marine food chains. These microscopic organisms, including diatoms, dinoflagellates, coccolithophores, and cyanobacteria, exhibit remarkable diversity in form and function while accounting for approximately 50% of global primary productivity despite their microscopic size. Diatoms exemplify the success of phytoplankton in marine environments, with their intricate silica cell walls creating some of nature's most beautiful microscopic structures while providing protection and regulating buoyancy. These organisms dominate phytoplankton communities in nutrient-rich waters and are particularly important in polar and coastal regions where their seasonal blooms support massive food webs. Dinoflagellates demonstrate the complexity that can exist within single-celled organisms, with many species possessing elaborate cell coverings, complex life cycles, and sophisticated behaviors including vertical migration and bioluminescence. Some dinoflagellates form harmful algal blooms that produce toxins affecting marine ecosystems and human health, while others engage in symbiotic relationships with coral and other marine invertebrates. Coccolithophores contribute to global carbon cycling through their unique ability to precipitate calcium carbonate plates, called coccoliths, which sink to the seafloor when the organisms die and contribute to marine sediments. These organisms are particularly sensitive to ocean acidification, which affects their ability to calcify and may have far-reaching implications for marine ecosystems and climate regulation. Cyanobacteria, also known as blue-green algae, include some of the ocean's smallest but most abundant photosynthetic organisms, with genera like Prochlorococcus and Synechococcus contributing enormously to marine primary productivity despite their tiny size. These organisms have streamlined genomes and simplified cellular structures that enable them to thrive in nutrient-poor open ocean environments. Zooplankton encompass a diverse array of heterotrophic organisms that feed on phytoplankton, other zooplankton, or organic particles, serving as crucial links between primary producers and higher trophic levels. This group includes both permanent members of the plankton community, such as copepods and krill, and temporary members such as the larval stages of many marine invertebrates and fish. Copepods represent the most abundant multicellular animals on Earth, with thousands of species filling diverse ecological roles from herbivorous grazers to predatory carnivores. These small crustaceans exhibit remarkable adaptations to planktonic life, including efficient swimming mechanisms, sophisticated sensory systems, and complex life cycles that allow them to exploit seasonal changes in food availability. Krill serve as keystone species in many marine ecosystems, particularly in polar regions where their massive swarms support populations of whales, seals, penguins, and other predators. These shrimp-like crustaceans undertake extensive vertical migrations and can form aggregations containing millions of individuals, creating some of the largest animal concentrations on Earth. Gelatinous zooplankton, including jellyfish, ctenophores, and salps, play increasingly recognized roles in marine food webs while demonstrating unique adaptations to planktonic existence. These organisms can achieve large sizes while maintaining near-neutral buoyancy through their high water content, and some species can rapidly increase their populations under favorable conditions. Marine microbial communities include bacteria, archaea, viruses, and protists that perform essential ecosystem functions, including nutrient cycling, decomposition, and primary production. These microscopic organisms process most of the organic matter in marine ecosystems and form complex food webs that operate largely independent of the traditional phytoplankton-zooplankton food chain. The microbial loop describes how dissolved organic matter released by phytoplankton and other organisms is processed by bacteria, which are then consumed by protist predators, ultimately transferring this material back into the traditional food web. This process is crucial for nutrient cycling and energy flow in marine ecosystems, particularly in oligotrophic open ocean environments. Viral ecology in marine systems involves complex interactions between viruses and their hosts that influence population dynamics, genetic diversity, and biogeochemical cycling. Marine viruses are extremely abundant, typically outnumbering bacteria by 10 to 1, and their infection and lysis of host cells releases nutrients and organic matter that fuel marine food webs. Planktonic life cycles often involve complex developmental stages and reproductive strategies that allow organisms to cope with environmental variability and exploit seasonal changes in conditions. Many species exhibit alternating between sexual and asexual reproduction, vertical migration patterns that track optimal conditions, and resting stages that allow survival through unfavorable periods. Seasonal plankton dynamics create predictable patterns of abundance and community composition that reflect changes in temperature, light, and nutrient availability. Spring phytoplankton blooms in temperate and polar regions support seasonal increases in zooplankton populations, which in turn affect fish reproduction and the timing of migration patterns in seabirds and marine mammals. Vertical migration of zooplankton represents one of the largest daily migrations on Earth, with billions of organisms moving up and down through the water column in response to light levels and predation pressure. This behavior affects the vertical distribution of nutrients and organic matter, while creating feeding opportunities for predators at different depths and times. Adaptation to planktonic life involves numerous morphological and physiological specializations that enable organisms to remain suspended in the water column, capture food efficiently, and avoid predation. These adaptations include reduced body density, elaborate appendages for swimming and feeding, transparent bodies for camouflage, and sophisticated sensory systems for detecting prey and predators. Harmful algal blooms represent situations where phytoplankton populations grow to levels that negatively impact marine ecosystems, human health, or economic activities. These events may be increasing in frequency and intensity due to nutrient pollution, climate change, and other human activities, highlighting the importance of understanding plankton ecology for marine resource management. Chapter 3: Coastal and Shallow Water Ecosystems. Coastal marine ecosystems represent some of the most productive and biologically diverse habitats on Earth, where the interface between terrestrial and marine environments creates unique conditions that support specialized communities of organisms. These dynamic systems are characterized by steep environmental gradients, high nutrient inputs, and frequent disturbance that collectively promote exceptional species diversity and ecosystem complexity. Rocky intertidal zones exemplify the challenges and opportunities present in coastal environments, where organisms must cope with the dramatic changes associated with tidal cycles that alternately expose them to marine and terrestrial conditions. The twice-daily rhythm of tidal exposure creates distinct vertical zonation patterns determined by each species' tolerance to desiccation, temperature variation, and wave action. Zonation patterns in rocky intertidal communities create distinct bands of organisms at different tidal heights, with splash zones dominated by hardy species like periwinkles and barnacles, mid-tide zones supporting diverse assemblages of mussels, barnacles, and algae, and low-tide zones harboring kelps, sea stars, and other species requiring constant submersion. These patterns reflect the fundamental trade-off between competitive ability and stress tolerance that shapes community structure. Wave action serves as a major structuring force in rocky intertidal ecosystems, with wave exposure determining which organisms can survive in different microhabitats. Exposed shores favor organisms with strong attachment mechanisms and streamlined body forms, while protected areas support more delicate species and complex three-dimensional community structures that would be destroyed by heavy wave action. Keystone predation effects are particularly well documented in rocky intertidal systems, where predators like sea stars can maintain community diversity by preventing competitive dominance from monopolizing space. The removal of these keystone predators often leads to dramatic shifts in community composition, demonstrating the crucial role of predation in maintaining ecosystem structure. Sandy beach ecosystems support specialized communities adapted to life in shifting sediments, where organisms must cope with wave action, sand abrasion, and limited attachment surfaces. The fauna is dominated by burrowing organisms, including polychaete worms, crustaceans, and mollusks, that can rapidly excavate burrows and feed on organic matter in the sediments or water column. Mudflat communities develop in protected coastal areas where fine sediments accumulate, creating habitats characterized by low oxygen conditions, high organic content, and extreme fluctuations in salinity and temperature. These environments support remarkable densities of deposit-feeding invertebrates that process enormous quantities of sediment, while serving as crucial feeding areas for migratory shorebirds. Salt marsh ecosystems represent the interface between terrestrial and marine environments, where specialized halophytic plants create primary productivity that supports complex food webs, including resident fish and invertebrates, as well as transient species that use these areas for feeding and nursery habitat. The regular tidal flooding creates dynamic conditions that challenge organisms while providing connectivity to adjacent marine systems. Seagrass beds form underwater meadows that rank among the most productive marine ecosystems, with flowering plants that have adapted to marine life, creating three-dimensional habitat structure that supports diverse communities of fish, invertebrates, and epiphytic organisms. These systems provide crucial nursery habitat for many commercially important fish species, while stabilizing sediments and improving water quality. Mangrove forests develop along tropical coastlines where salt-tolerant trees create unique ecosystems characterized by complex prop root systems that provide habitat for diverse marine organisms while protecting coastlines from storm damage. The detritus produced by mangrove trees supports extensive food webs that include both resident species and migrants from adjacent marine environments. Coral reef ecosystems represent the marine equivalent of tropical rainforests, supporting extraordinary biodiversity in nutrient-poor tropical waters through the symbiotic relationship between coral polyps and zooxanthellae algae. The calcium carbonate skeletons secreted by corals create complex three-dimensional structures that provide habitat for thousands of species, while forming barriers that protect coastlines from wave erosion. Coral biology involves the intimate partnership between animal hosts and photosynthetic symbionts that enables reef-building corals to thrive in nutrient-poor waters while secreting massive limestone structures. This symbiosis is highly sensitive to environmental stress, with elevated temperatures, pollution, and ocean acidification causing coral bleaching events that threaten reef ecosystems worldwide. Reef fish communities exhibit remarkable diversity and specialization, with different species occupying distinct ecological niches defined by feeding behavior, habitat preferences, and social organization. Cleaner fish remove parasites from larger species, herbivorous fish control algal growth on reefs, and predatory fish regulate populations of smaller species, creating complex interaction networks that maintain reef ecosystem function. Kelp forest ecosystems develop in cold, nutrient-rich waters, where large brown algae create underwater forests that support diverse communities of marine organisms. These highly productive systems are structured by the canopy-forming kelps that modify light and flow conditions, while providing habitat and food for numerous species of fish, invertebrates, and marine mammals. Estuarine environments where rivers meet the sea create dynamic ecosystems characterized by fluctuating salinity, temperature, and nutrient levels that challenge organisms while providing exceptional productivity. The mixing of fresh and salt water creates unique physical and chemical conditions that trap nutrients and support some of the most productive ecosystems on Earth. Tidal pool communities represent miniature ecosystems that develop in depressions on rocky shores, where small volumes of seawater, trapped at low tide, create isolated habitats with extreme environmental conditions. These pools serve as refuges for mobile organisms during low tide, while harboring specialized communities adapted to the fluctuating conditions. Coastal upwelling zones create some of the most productive marine ecosystems where wind-driven upwelling brings nutrient-rich deep water to the surface, supporting massive phytoplankton blooms that form the base of productive food webs. These systems support major fisheries, while demonstrating the importance of physical oceanographic processes in controlling biological productivity. Human impacts on coastal ecosystems include habitat destruction, pollution, overfishing, and climate change effects that threaten the integrity of these productive and diverse systems. Coastal development has destroyed vast areas of salt marshes, mangroves, and other critical habitats, while nutrient pollution has created dead zones and harmful algal blooms that degrade ecosystem function. Conservation of coastal ecosystems requires integrated management approaches that address the multiple stressors affecting these systems, while recognizing their crucial roles in supporting marine biodiversity, protecting coastlines, and providing ecosystem services to human communities. Marine protected areas, habitat restoration, and pollution control represent important tools for coastal conservation. Chapter 4: Deep Sea and Open Ocean Environments. The deep sea represents Earth's largest habitat, encompassing the vast volumes of water below the continental shelf and the seafloor in areas deeper than 200 meters where sunlight cannot penetrate. This realm of perpetual darkness, extreme pressure, and near-freezing temperatures supports unique communities of organisms that have evolved remarkable adaptations to survive in one of our planet's most challenging environments. Pelagic zonation divides the open ocean into distinct depth zones that reflect changes in physical conditions and biological communities, with each zone characterized by specific environmental conditions and associated fauna. The epipelagic zone supports most of the ocean's primary productivity and largest animals, while deeper zones become increasingly food-limited and specialized for life under extreme conditions. The mesopelagic zone, extending from approximately 200 to 1,000 meters depth, harbors the largest animal migration on Earth, as countless organisms undertake daily vertical migrations between the depths where they spend daylight hours and surface waters where they feed at night. This twilight zone supports enormous biomass, despite receiving no direct sunlight, relying instead on the rain of organic matter from surface waters and the daily influx of migrating organisms. Bathypelagic waters below 1,000 meters depth represent the true deep sea, where temperatures approach freezing, pressure exceeds 100 atmospheres, and food is extremely scarce. The organisms living in this zone exhibit extraordinary adaptations, including bioluminescence, expandable jaws and stomachs, and energy-efficient lifestyles that enable survival in this food-limited environment. Abyssal plains covering vast areas of the deep seafloor appear deceptively barren, but actually support diverse communities of organisms adapted to life in soft sediments under extreme pressure. The fauna includes deposit-feeding invertebrates, scavenging crustaceans, and specialized fish that have evolved unique feeding strategies to exploit the limited food resources available in this environment. Hydrothermal vent ecosystems represent oases of life in the deep sea, where chemical energy from Earth's interior supports productive communities based on chemosynthetic bacteria rather than photosynthetic primary production. These remarkable ecosystems harbor unique fauna including giant tube worms, eyeless shrimp, and specialized fish that have evolved to thrive in conditions of extreme temperature, pressure, and chemical toxicity. Cold seep communities develop where hydrocarbons and hydrogen sulfide seep from the seafloor, creating chemosynthetic ecosystems that can persist for decades or centuries, compared to the ephemeral nature of hydrothermal vents. These systems support communities dominated by bivalves, tube worms, and bacterial mats that derive energy from the oxidation of methane and hydrogen sulfide. Seamount ecosystems create islands of hard substrate in the deep sea that support unique communities, often characterized by high levels of endemism. These underwater mountains alter ocean currents and create upwelling that enhances local productivity, while providing habitat for sessile invertebrates that require hard surfaces for attachment. Deep-sea adaptations reflect the extreme conditions of pressure, darkness, and food scarcity that characterize this environment. Organisms have evolved specialized pressure-resistant proteins, bioluminescent communication systems, energy-efficient metabolisms, and anatomical modifications that enable them to function under conditions that would be lethal to most surface-dwelling organisms. Bioluminescence serves multiple functions in deep-sea organisms, including communication, prey attraction, predator deterrence, and camouflage through counterlumination. The biochemical mechanisms of light production have evolved independently in numerous deep-sea lineages, creating a diverse array of light-producing systems using different chemical reactions and anatomical structures. Deep-sea feeding strategies reflect the extreme scarcity of food in most deep-sea environments, with organisms exhibiting adaptations for capturing rare prey items, processing carrion that sinks from surface waters, and maximizing energy extraction from limited food sources. Many deep-sea predators have expandable jaws and stomachs that allow them to consume prey larger than themselves. Vertical migration patterns connect surface and deep-sea ecosystems through the daily movements of organisms that feed in productive surface waters, but spend daylight hours in the safety of deeper waters. This migration transfers enormous quantities of carbon and nutrients from surface waters to the deep sea, while supporting deep-sea predators that intercept migrating organisms. Deep-sea reproduction often involves adaptations to the challenges of finding mates in a vast, sparsely populated environment. Some species exhibit sexual dimorphism with tiny males that become parasitic on much larger females, while others produce bioluminescent mating displays or chemical attractants that increase the probability of encountering potential mates. Abyssal gigantism describes the tendency for some deep-sea organisms to reach much larger sizes than their shallow water relatives, possibly reflecting reduced predation pressure, longevity, or other factors associated with deep-sea environments. Examples include giant isopods, amphipods, and sea spiders that can reach remarkable sizes in the deep sea. Deep-sea biodiversity patterns differ from those in surface waters, with some areas supporting unexpectedly high species diversity, despite the challenging environmental conditions. The deep sea may harbor more species than previously recognized, with new species being discovered regularly as exploration technology advances and previously inaccessible areas are studied. Connectivity between deep-sea populations can be maintained across vast distances through larval dispersal in deep water currents, though the mechanisms and scales of this connectivity remain poorly understood. Understanding population connectivity is crucial for designing effective conservation strategies for deep-sea ecosystems that may be threatened by mining and other human activities. Deep-sea mining represents an emerging threat to these unique ecosystems, as technology advances to enable extraction of polymetallic nodules, sulfide deposits, and other mineral resources from the deep seafloor. The potential impacts of mining on deep-sea communities that may require decades or centuries to recover from disturbance are not well understood. Climate change effects on deep-sea ecosystems include ocean warming, acidification, and deoxygenation that may alter the distribution and survival of deep-sea organisms. These changes may be particularly significant for organisms adapted to the stable conditions that have characterized the deep sea for millions of years. Exploration technologies, including remotely operated vehicles, autonomous underwater vehicles, and advanced camera systems, are revolutionizing our understanding of deep-sea ecosystems by enabling direct observation and sampling of previously inaccessible environments. These tools are revealing the complexity and diversity of deep-sea life while providing insights into ecosystem functioning in Earth's largest habitat. Chapter 5: Marine Conservation and Human Impacts. The relationship between human activities and marine ecosystems has become increasingly complex and concerning as our population grows and our technological capabilities expand, creating unprecedented pressures on ocean environments that have evolved over millions of years. Understanding these impacts and developing effective conservation strategies represents one of the most critical challenges facing marine biology and society as a whole. Overfishing represents perhaps the most pervasive human impact on marine ecosystems, with industrial fishing operations capable of removing entire populations of target species while causing significant mortality of non-target species through bycatch. The collapse of major fisheries worldwide demonstrates how intensive exploitation can overwhelm the reproductive capacity of marine species, leading to ecosystem-wide changes that may persist for decades even after fishing pressure is reduced. Marine food web alterations result from selective removal of top predators, herbivores, and other key species through fishing, creating cascading effects that propagate through entire ecosystems. The removal of large predatory fish can lead to increases in their prey species, which may then overconsume their own food sources, ultimately affecting primary producers and ecosystem structure in ways that were not anticipated when fishing began. Habitat destruction in marine environments includes physical damage from fishing gear, coastal development, dredging, and other activities that destroy or degrade the three-dimensional structure that many marine organisms require for shelter, feeding, and reproduction. Bottom trawling, in particular, can destroy complex benthic communities that may require decades or centuries to recover. Pollution in marine environments encompasses a vast array of contaminants including nutrients, chemicals, plastics, oil, and other substances that can affect marine organisms through direct toxicity, habitat degradation, or bioaccumulation through food webs. Marine plastic pollution has become a particularly visible problem, with microplastics now found throughout marine food webs and in the tissues of organisms from plankton to top predators. Ocean acidification, caused by the absorption of atmospheric carbon dioxide, is altering the chemistry of seawater in ways that affect the ability of many marine organisms to build shells, skeletons, and other calcium carbonate structures. This process is particularly threatening to coral reefs, shellfish, and other calcifying organisms that form the foundation of many marine ecosystems. Climate change impacts on marine ecosystems include rising temperatures, changing precipitation patterns, sea level rise, and altered ocean circulation that are causing shifts in species distributions, breeding cycles, and ecosystem productivity. Many marine species are moving toward polar regions or deeper waters in response to warming, creating novel community assemblages and potentially disrupting existing ecological relationships. Coral bleaching events caused by elevated water temperatures have become increasingly frequent and severe, with some reefs experiencing multiple bleaching events within a few years that prevent recovery and lead to widespread coral mortality. The loss of coral reefs affects not only the extraordinary biodiversity they support, but also the coastal protection and economic benefits they provide to human communities. Marine protected areas represent one of the most important tools for marine conservation, providing refuges where marine organisms can reproduce and grow without direct human exploitation. Effective marine protected areas can restore populations of exploited species, protect critical habitats, and maintain ecosystem processes that benefit both conservation and fisheries management goals. Fisheries management has evolved from simple exploitation models to ecosystem-based approaches that consider the broader ecological context of target species and the cumulative impacts of fishing on marine ecosystems. This evolution reflects growing recognition that sustainable fisheries require maintaining the integrity of marine food webs and habitats, not just managing individual species. Marine restoration involves active efforts to repair damaged marine ecosystems through activities such as coral reef restoration, seagrass replanting, oyster reef reconstruction, and habitat enhancement. While these efforts can be successful on local scales, they require substantial resources and long-term commitment to achieve meaningful conservation benefits. Invasive species introduction through shipping, aquaculture, and other human activities can dramatically alter marine ecosystems by introducing organisms that lack natural predators or competitors in their new environments. Some marine invasions have caused major ecological and economic damage, highlighting the importance of prevention and early detection in managing this threat. Pollution prevention and remediation efforts include regulations to control point source pollution, best management practices to reduce non-point source pollution, and cleanup technologies to address contaminated marine environments. The success of these efforts varies widely depending on the type of pollution, the scale of contamination, and the commitment of regulatory agencies and stakeholders. International cooperation in marine conservation is essential because ocean ecosystems cross political boundaries, and many marine species are highly migratory, requiring coordinated management efforts among multiple countries. International agreements and organizations play crucial roles in coordinating conservation efforts and establishing standards for marine resource management. Marine ecosystem services encompass the many benefits that healthy marine ecosystems provide to human society, including food production, climate regulation, coastal protection, waste processing, recreation, and cultural values. Understanding and quantifying these services helps demonstrate the economic importance of marine conservation while providing frameworks for incorporating ecosystem values into decision-making processes. Community-based conservation recognizes that local communities often have strong incentives to maintain healthy marine resources and may be more effective at enforcement and long-term stewardship than distant government agencies. Successful community-based conservation programs combine traditional ecological knowledge with scientific understanding while providing economic benefits to participating communities. Marine spatial planning involves systematic approaches to allocating ocean space among competing uses while maintaining ecosystem integrity and services. This process requires balancing conservation needs with economic activities such as fishing, shipping, energy development, and recreation in ways that minimize conflicts and maximize benefits for both human communities and marine ecosystems. Emerging threats to marine ecosystems include deep-sea mining, ocean geoengineering, noise pollution, and new forms of contamination that may have unpredictable consequences for marine life. Addressing these threats requires proactive research and precautionary management approaches that can prevent problems rather than simply responding to them after they occur. Technology applications in marine conservation include satellite monitoring of fishing activities, genetic tools for tracking species and populations, autonomous vehicles for ecosystem monitoring, and modeling systems that can predict the effects of different management scenarios. These technologies are expanding our ability to understand and manage marine ecosystems while reducing the costs of conservation and monitoring activities. Education and outreach play crucial roles in marine conservation by building public understanding of marine ecosystems and support for conservation measures. Effective communication about marine conservation challenges and solutions can influence individual behavior, support for policy changes, and participation in conservation activities that collectively contribute to protecting marine biodiversity. The future of marine conservation will depend on integrating scientific understanding with effective governance, economic incentives, and social engagement to address the multiple stressors affecting marine ecosystems. Success will require unprecedented cooperation among scientists, policymakers, industry, and the public to develop and implement solutions that can maintain healthy marine ecosystems while supporting human needs and aspirations.
Chapter 1. The Cellular Foundation of Plant Life. Plant physiology represents the study of how plants function at the most fundamental levels, from the molecular processes occurring within individual cells to the integrated responses of whole organisms to their environment. Understanding plant physiology requires examining the unique cellular architecture and biochemical pathways that enable plants to capture energy from sunlight, extract nutrients from soil, and coordinate growth and development throughout their lives. At its core, plant physiology reveals how these remarkable organisms have evolved sophisticated mechanisms to thrive as stationary beings that must obtain all their resources from their immediate surroundings. The plant cell serves as the fundamental unit of plant life, distinguished from animal cells by several unique features that directly relate to plant physiological processes. The presence of a rigid cell wall composed primarily of cellulose provides structural support that enables plants to grow tall and compete effectively for light, while also creating unique challenges for cell expansion and intercellular communication. This cell wall represents far more than a simple protective barrier, functioning as a dynamic structure that responds to environmental cues, facilitates water and nutrient transport, and provides defense against pathogens and herbivores. Within the cell wall lies the plasma membrane, a selectively permeable barrier that controls the movement of substances into and out of the cell. Plant plasma membranes contain specialized transport proteins that facilitate the uptake of essential nutrients while excluding potentially harmful compounds. These membrane systems exhibit remarkable specificity and regulation, allowing plants to maintain precise control over their internal chemical environment despite dramatic fluctuations in external conditions. The chloroplast stands as perhaps the most distinctive organelle of plant cells, housing the remarkable machinery of photosynthesis that converts light energy into chemical energy. These complex organelles contain their own DNA and ribosomes, reflecting their evolutionary origin as endosymbiotic cyanobacteria that formed partnerships with early eukaryotic cells billions of years ago. The internal structure of chloroplasts, with their elaborate thylakoid membrane systems and surrounding stroma, creates specialized compartments that optimize the light-dependent and light-independent reactions of photosynthesis. Chlorophyll molecules embedded within thylakoid membranes capture light energy and transfer it through complex electron transport chains that generate the chemical energy carriers ATP and NADPH. The stroma surrounding the thylakoids contains the enzymes responsible for carbon fixation, where atmospheric carbon dioxide is incorporated into organic molecules through the Calvin cycle. This compartmentalization allows plants to separate the potentially damaging light reactions from the more stable carbon fixation processes, while maintaining efficient energy transfer between these interconnected systems. The vacuole represents another distinctive feature of plant cells, often occupying the majority of the cell's volume in mature tissues. Far from being simply an empty space, the vacuole serves multiple crucial physiological functions, including maintaining cell turgor pressure, storing nutrients and metabolic waste products, and providing structural support. The vacuolar membrane, or tonoplast, contains numerous transport proteins that regulate the movement of ions and organic compounds between the vacuole and cytoplasm, playing crucial roles in cellular homeostasis and plant responses to environmental stress. Turgor pressure generated by water uptake into the vacuole provides the driving force for cell expansion and maintains the structural integrity of non-woody plant tissues. Changes in turgor pressure enable plant movements such as the opening and closing of stomata, the rapid responses of sensitive plant leaves, and the diurnal movements of flowers and leaves that track the sun's position. The regulation of turgor pressure through controlled water and solute movement represents one of the most fundamental aspects of plant physiology. The endoplasmic reticulum and Golgi apparatus in plant cells perform functions similar to those in animal cells, synthesizing and processing proteins and other cellular components. However, plant cells also contain unique organelles such as peroxisomes that play specialized roles in photorespiration and lipid metabolism. These organelles work in concert with chloroplasts and mitochondria to maintain cellular energy balance and metabolic homeostasis under varying environmental conditions. Mitochondria in plant cells serve dual roles, functioning both as sites of cellular respiration similar to animal cells and as crucial components of photorespiration in photosynthetic tissues. Plant mitochondria exhibit unique biochemical features, including alternative oxidase pathways that allow continued respiration under stress conditions when the normal electron transport chain may be inhibited. This metabolic flexibility provides plants with remarkable resilience to environmental challenges that might severely impact organisms dependent solely on conventional respiratory pathways. The cytoskeleton of plant cells, composed of microtubules, microfilaments, and intermediate filaments, plays crucial roles in cell division, organelle movement, and the coordination of cellular activities. Microtubules are particularly important in plant cells, guiding the deposition of cellulose fibers in the cell wall and determining the direction of cell expansion. The dynamic reorganization of cytoskeletal elements allows plant cells to respond rapidly to environmental cues and coordinate developmental processes throughout the organism. Plasmodesmata represent unique intercellular connections that distinguish plant tissues from animal tissues, creating cytoplasmic continuities between adjacent cells that facilitate communication and transport. These microscopic channels allow the movement of water, ions, small molecules, and even larger proteins and RNA molecules between cells, creating a coordinated network that enables rapid signaling throughout plant tissues. The regulation of plasmodesmata permeability provides plants with sophisticated mechanisms for controlling intercellular transport and coordinating physiological responses. Plant cell walls exhibit remarkable diversity in composition and structure, with different cell types producing walls optimized for their specific functions. Primary cell walls in growing tissues remain relatively thin and flexible to accommodate cell expansion, while secondary cell walls in mature tissues become thick and rigid to provide structural support. The composition of cell walls varies among different plant species and tissue types, with specialized components such as lignin in woody tissues, suberin in protective tissues, and cutin in epidermal surfaces providing unique functional properties. The synthesis and modification of cell wall components represents a major metabolic investment for plants, often accounting for a significant portion of the carbon fixed through photosynthesis. Plants must carefully balance the allocation of resources between cell wall synthesis, growth processes, and other metabolic requirements, while responding to environmental challenges that may require rapid adjustments in cell wall properties. Understanding these regulatory mechanisms provides insights into how plants optimize their growth and survival under varying conditions. Cellular communication systems in plants rely heavily on chemical signaling molecules that coordinate physiological processes throughout the organism. Plant hormones such as auxin, gibberellins, cytokinins, abscisic acid, and ethylene serve as key regulatory molecules that influence virtually every aspect of plant development and environmental response. These signaling systems operate through complex networks of synthesis, transport, perception, and response mechanisms that enable plants to integrate information from multiple sources and coordinate appropriate physiological adjustments. The transport of signaling molecules within plant cells and between different tissues requires sophisticated delivery systems that can operate over both short and long distances. Some signals move through the symplastic pathway via plasmodesmata, while others utilize the apoplastic pathway through cell walls and intercellular spaces. Long-distance signaling often involves the vascular system, where both chemical and electrical signals can be transmitted rapidly throughout the plant body. Modern techniques in plant cell biology have revealed the extraordinary complexity and sophistication of plant cellular systems, challenging earlier views of plants as relatively simple organisms. Advanced microscopy techniques, molecular biology methods, and computational approaches continue to unveil new aspects of plant cellular function and regulation. These discoveries have profound implications for understanding how plants respond to environmental challenges and for developing strategies to improve crop productivity and stress tolerance in agricultural systems. Chapter 2: Photosynthesis and Energy Conversion. Photosynthesis stands as one of the most important biological processes on Earth, representing the primary mechanism by which solar energy is converted into chemical energy that powers virtually all life on our planet. This remarkable process enables plants to capture light energy and use it to synthesize organic molecules from simple inorganic compounds, creating the foundation for all food webs while simultaneously producing the oxygen that most organisms require for survival. Understanding the intricate mechanisms of photosynthesis reveals how plants have evolved sophisticated molecular machinery to efficiently harvest light energy and convert it into stable chemical forms that can be stored and utilized for growth, reproduction, and survival. The overall process of photosynthesis can be summarized by the deceptively simple equation: 6 carbon dioxide plus 6 hydrogen oxide plus light energy right pointing arrow C, hydrogen oxide plus 6O. However, this equation masks the extraordinary complexity of the underlying biochemical pathways that involve hundreds of different molecules working in precisely coordinated sequences. Photosynthesis consists of two interconnected sets of reactions: the light-dependent reactions that capture and convert light energy into chemical energy, and the light-independent reactions that use this chemical energy to fix atmospheric carbon dioxide into organic molecules. The light-dependent reactions occur within the thylakoid membranes of chloroplasts, where specialized protein complexes, called photosystems, capture light energy and convert it into chemical energy in the form of ATP and NADPH. These photosystems contain hundreds of chlorophyll and other pigment molecules arranged in precise geometric patterns that optimize light absorption and energy transfer. The arrangement of these pigments creates antenna complexes that funnel light energy to reaction centers where the actual conversion of light energy to chemical energy occurs. Photosystem 2 represents the beginning of the light-dependent reactions, containing a reaction center chlorophyll molecule that absorbs light energy and becomes excited to a higher energy state. This excited chlorophyll molecule donates an electron to a primary electron acceptor, beginning an electron transport chain that ultimately generates ATP through a process called photophosphorylation. The electron lost from photosystem 2 is replaced by electrons extracted from water molecules in a remarkable process that splits water and releases oxygen as a byproduct. The water-splitting reaction catalyzed by photosystem 2 represents one of the most challenging chemical processes in biology, requiring the removal of 4 electrons from 2 water molecules to produce 1 oxygen molecule. This reaction is facilitated by a manganese-calcium cluster within the photosystem that accumulates the energy required to break the strong bonds in water molecules. The evolution of this water-splitting capability was crucial for the development of oxygenic photosynthesis and fundamentally changed Earth's atmosphere and the course of biological evolution. Electrons from photosystem 2 flow through a series of electron carriers including plastoquinone, cytochrome B6F complex, and plastocyanin before reaching photosystem. This electron transport chain is coupled to the pumping of protons across the thylakoid membrane, creating a proton gradient that drives ATP synthesis through ATP synthase, a remarkable molecular motor that converts the energy stored in proton gradients into the chemical bonds of ATP molecules. Photosystem I captures additional light energy and uses it to further energize electrons, ultimately transferring them to NADP to form NADPH. The cyclic and non-cyclic electron flow pathways allow plants to adjust the ratio of ATP to NADPH production depending on their metabolic needs and environmental conditions. This flexibility in energy conversion provides plants with the ability to optimize their photosynthetic efficiency under varying light conditions and metabolic demands. The light-independent reactions, also known as the Calvin cycle, occur in the stroma of chloroplasts and utilize the ATP and NADPH produced by the light-dependent reactions to fix atmospheric carbon dioxide into organic molecules. The key enzyme in this process, ribulose 1,5-bisphosphate carboxylase, oxygenase, Rubisco, catalyzes the addition of carbon dioxide to ribulose 1,5-bisphosphate, beginning a complex series of reactions that ultimately produce glucose and regenerate the starting materials for continued carbon fixation. Rubisco represents both the most abundant protein on Earth and one of the most important enzymes for life, yet it exhibits relatively poor specificity and efficiency compared to many other enzymes. This enzyme can react with both carbon dioxide and O, with the oxygenase reaction leading to photorespiration, a seemingly wasteful process that can significantly reduce photosynthetic efficiency. The dual specificity of Rubisco reflects evolutionary constraints and the changing atmospheric composition over geological time, as the enzyme evolved when atmospheric carbon dioxide concentrations were much higher and O concentrations were lower than current levels. The Calvin cycle requires careful regulation to balance carbon fixation with the regeneration of ribulose 1,5-bisphosphate and to coordinate carbon metabolism with the availability of ATP and NADPH from the light-dependent reactions. This regulation involves multiple mechanisms, including enzyme activation and deactivation, metabolite-based feedback inhibition, and light-dependent regulatory systems that ensure carbon fixation occurs primarily during daylight hours when energy is available from the light-dependent reactions. Photorespiration occurs when Rubisco reacts with oxygen instead of carbon dioxide, leading to the production of compounds that must be metabolized through a complex pathway involving chloroplasts, peroxisomes, and mitochondria. While photorespiration reduces the net efficiency of photosynthesis, it may serve important protective functions under stress conditions and plays roles in nitrogen metabolism and other cellular processes. The rate of photorespiration increases with temperature and decreases with carbon dioxide concentration, making it particularly problematic for plants in hot, dry environments. Some plants have evolved alternative photosynthetic pathways that minimize photorespiration and improve photosynthetic efficiency under specific environmental conditions. C4 photosynthesis, found in many tropical grasses including maize and sugarcane, concentrates carbon dioxide around Rubisco through spatial separation of initial carbon fixation and the Calvin cycle. This process requires additional energy but virtually eliminates photorespiration and allows efficient photosynthesis even under hot, dry conditions. CAM, crassylation acid metabolism, photosynthesis represents another adaptation to arid environments, involving temporal rather than spatial separation of carbon dioxide uptake and fixation. CAM plants open their stomata at night to take up carbon dioxide, which is stored as organic acids and then released during the day when stomata are closed to conserve water. This strategy allows plants to photosynthesize while minimizing water loss, though it typically results in slower growth rates compared to C3 and C4 plants. The efficiency of photosynthesis is influenced by numerous environmental factors, including light intensity, light quality, temperature, carbon dioxide concentration, and water availability. Light saturation curves reveal that photosynthetic rates increase with light intensity up to a maximum beyond which additional light provides no benefit and may even cause photoinhibition. Different plant species exhibit different light saturation points, reflecting their adaptations to specific light environments. Temperature affects photosynthesis through its impacts on enzyme activity, membrane fluidity, and the balance between carbon dioxide and O reactions of Rubisco. Most plants exhibit optimal temperatures for photosynthesis that reflect their evolutionary history and typical growing conditions. Extreme temperatures can damage the photosynthetic apparatus, particularly the protein complexes involved in light harvesting and electron transport. Water stress affects photosynthesis primarily through its impact on stomatal conductance, as plants close their stomata to conserve water, thereby reducing carbon dioxide uptake and limiting carbon fixation. Severe water stress can also directly damage chloroplasts and reduce the efficiency of photosynthetic processes. The coordination between photosynthesis and water use represents a fundamental constraint that shapes plant evolution and ecology. Atmospheric carbon dioxide concentration directly affects the rate of carbon fixation, with most plants showing increased photosynthetic rates under elevated carbon dioxide conditions. However, this carbon dioxide fertilization effect often diminishes over time due to acclimation responses and limitations imposed by other factors such as nitrogen availability. Understanding plant responses to changing atmospheric carbon dioxide concentrations is crucial for predicting how vegetation will respond to ongoing climate change. The regulation of photosynthesis involves complex feedback mechanisms that coordinate light harvesting, electron transport, and carbon fixation with overall plant metabolism and environmental conditions. These regulatory systems operate on multiple timescales, from rapid adjustments in enzyme activity that occur within seconds to longer-term changes in gene expression and chloroplast development that occur over hours to days. Photoinhibition represents an important protective mechanism that prevents damage to the photosynthetic apparatus under high light conditions. When light energy exceeds the capacity for productive photosynthesis, excess energy can be dissipated as heat through non-photochemical quenching mechanisms that protect against oxidative damage. Plants also possess repair mechanisms that can replace damaged components of the photosynthetic apparatus, though these repair processes require energy and resources that could otherwise be used for growth and reproduction. Modern research in photosynthesis continues to reveal new aspects of this fundamental process, with implications for understanding plant evolution, ecology, and responses to environmental change. Advances in structural biology have provided detailed views of photosynthetic protein complexes, while molecular biology techniques have enabled detailed studies of photosynthetic regulation and the evolution of different photosynthetic pathways. These discoveries are informing efforts to improve crop photosynthetic efficiency and develop plants better adapted to changing environmental conditions. Chapter 3: Water Relations and Transport. Water represents the most critical resource for plant survival and function, serving as the medium for virtually all biochemical reactions, the primary component of plant structure, and the driving force for nutrient transport throughout the plant body. Understanding plant-water relations requires examining the complex physical and physiological mechanisms that govern water uptake, transport, and loss, revealing how plants have evolved sophisticated strategies to maintain water balance while meeting their metabolic needs and responding to environmental challenges. The movement of water through plants follows fundamental physical principles while being precisely regulated by biological mechanisms that coordinate water transport with photosynthesis, growth, and environmental responses. The journey of water through plants begins in the soil, where water exists in various forms depending on soil texture, organic matter content, and moisture conditions. Plants can readily absorb water that exists as a thin film around soil particles or fills larger pore spaces, but much of the water in soil may be unavailable due to strong adhesive forces that bind it to soil particles or osmotic effects of dissolved salts. The availability of soil water depends not only on total moisture content, but also on soil water potential, which determines the energy required for plants to extract water from soil. Water uptake by plant roots occurs primarily through specialized regions where root hairs dramatically increase the surface area available for absorption. These microscopic extensions of root epidermal cells can increase the absorptive surface area by 10 to 40 times, enabling plants to efficiently extract water from soil. Root hairs are particularly abundant in young, actively growing root tissues and may be absent from older, suberized root sections that serve primarily for transport and structural support. The process of water uptake involves movement across multiple cellular barriers, beginning with passage through the root epidermis and continuing through the cortex toward the central vascular tissues. Water can follow two primary pathways during this journey: the apoplastic pathway through cell walls and intercellular spaces, and the symplastic pathway through living cell cytoplasm connected by plasmodesmata. The relative importance of these pathways varies depending on root development, environmental conditions, and the presence of barriers such as the Casparian strip. The Casparian strip represents a crucial regulatory barrier that controls water and solute movement into the root vascular system. This band of hydrophobic material in the cell walls of endodermal cells forces water and dissolved substances to pass through cell membranes rather than moving freely through cell walls, enabling plants to selectively control which substances enter the vascular system. This selective barrier prevents the passive movement of potentially harmful substances while ensuring that essential nutrients are actively transported into the plant. Once water enters the root vascular system, it moves through the xylem tissue, which consists of specialized conducting cells that form continuous pathways from roots to leaves. The xylem contains two main types of conducting elements: tracheids, which are elongated cells with tapered ends connected through pits in their cell walls, and vessel elements, which are shorter, wider cells with perforated end walls that form continuous tubes called vessels. These conducting elements are dead at maturity, forming hollow pipelines that offer minimal resistance to water flow. The driving force for water movement through the xylem comes primarily from transpiration, the process by which water evaporates from leaf surfaces and creates negative pressure that pulls water upward through the plant. This transpiration cohesion tension mechanism relies on the remarkable cohesive properties of water molecules, which form hydrogen bonds that allow continuous water columns to withstand the negative pressures required to lift water to great heights. The strength of these water columns is truly remarkable, with tensions often exceeding 10 atmospheres in tall trees. Transpiration occurs primarily through stomata, microscopic pores in leaf surfaces that are surrounded by specialized guard cells capable of opening and closing the pores. These guard cells respond to multiple environmental and internal signals, including light, carbon dioxide concentration, humidity, temperature, and plant water status. The regulation of stomatal aperture represents one of the most important mechanisms by which plants control their water balance, while maintaining adequate carbon dioxide uptake for photosynthesis. The opening and closing of stomata involves complex changes in guard cell turgor pressure, driven by the uptake and loss of potassium ions and other osmotically active substances. When stomata open, guard cells accumulate potassium and other ions, causing water uptake and increased turgor pressure that changes cell shape and creates the stomatal pore. Stomatal closure involves the reverse process, with ion efflux leading to water loss and decreased turgor pressure that closes the pore. The coordination between stomatal behavior and photosynthesis represents a fundamental challenge for plants, as the same pores that allow carbon dioxide uptake also permit water loss. Plants must carefully balance the need for carbon dioxide with the need to conserve water, particularly under drought conditions where excessive water loss could threaten survival. This balance is achieved through sophisticated regulatory mechanisms that integrate information about light availability, atmospheric carbon dioxide concentration, plant water status, and other environmental factors. Different plant species have evolved various strategies for optimizing water use efficiency, defined as the amount of carbon fixed per unit of water lost through transpiration. Some plants exhibit high water use efficiency through specialized photosynthetic pathways, reduced stomatal conductance, or structural adaptations that minimize water loss. Others prioritize rapid growth and high photosynthetic rates, even at the cost of greater water consumption, reflecting different evolutionary strategies for different environments. The measurement of plant water status involves several important parameters that describe different aspects of water relations. Water potential represents the chemical potential of water in plant tissues compared to pure water, providing a unified measure that accounts for both pressure and osmotic effects. Turgor pressure describes the pressure exerted by cell contents against cell walls, while osmotic potential reflects the concentration of dissolved substances that affect water movement. Plants exhibit various responses to water stress that help them survive periods of limited water availability. Short-term responses include stomatal closure to reduce water loss, changes in leaf orientation to minimize solar radiation absorption, and adjustments in osmotic potential to maintain turgor pressure. Longer-term responses may include changes in root growth to explore new soil volumes, modifications in leaf structure to reduce transpiration, and alterations in metabolic processes to conserve energy and resources. Root system architecture plays a crucial role in plant-water relations, with different species exhibiting various strategies for water acquisition. Deep root systems allow access to groundwater or soil moisture at depth, while extensive shallow root systems can rapidly capture precipitation before it evaporates or moves beyond the root zone. Some plants exhibit dimorphic root systems that combine both strategies, maximizing their ability to acquire water under different moisture conditions. The hydraulic architecture of plants involves trade-offs between efficient water transport and safety from hydraulic failure. Wider conducting elements transport water more efficiently, but are more vulnerable to cavitation, the formation of air bubbles that can block water transport. Plants in different environments have evolved different hydraulic strategies, with those in drought-prone areas typically exhibiting more conservative hydraulic architectures that prioritize safety over efficiency. Aquaporins represent a recently discovered class of water channel proteins that facilitate rapid water movement across cell membranes. These proteins can dramatically increase membrane water permeability and are subject to complex regulation that allows plants to fine-tune water transport at the cellular level. Different types of aquaporins are found in different tissues and respond to various environmental and developmental signals, providing plants with sophisticated mechanisms for controlling water movement. The integration of water transport with nutrient transport represents another important aspect of plant physiology, as the xylem stream carries dissolved minerals from roots to leaves, while the phloem transports organic compounds throughout the plant. The coordination between these transport systems ensures that water movement supports both the delivery of essential nutrients and the redistribution of photosynthetic products to growing and storage tissues. Climate change is altering precipitation patterns and increasing temperatures worldwide, creating new challenges for plant-water relations. Understanding how plants respond to changing water availability and developing crops with improved drought tolerance represents a critical challenge for agriculture and natural resource management. Research in plant-water relations continues to reveal new mechanisms and strategies that may inform efforts to develop more water-efficient crops and conservation strategies. Modern techniques for studying plant-water relations include sophisticated instrumentation for measuring water potential, sap flow, and stomatal conductance, as well as molecular approaches for understanding the regulation of water transport proteins and stress response pathways. These tools are providing new insights into the mechanisms underlying plant-water relations and informing strategies for improving plant performance under water-limited conditions. Chapter 4: Plant Nutrition and Mineral Transport. Plant nutrition encompasses the complex processes by which plants obtain, transport, and utilize the essential mineral elements required for growth, development, and physiological function. Understanding plant nutrition requires examining not only which elements are essential and why they are needed, but also the sophisticated mechanisms that enable plants to acquire these nutrients from soil solutions, transport them throughout the plant body, and regulate their uptake and utilization in response to availability and metabolic demands. This field reveals how plants have evolved remarkable strategies to thrive in environments where essential nutrients may be scarce, unevenly distributed, or present in forms that are difficult to access. Essential plant nutrients are traditionally divided into macronutrients, which are required in relatively large quantities, and micronutrients, which are needed in much smaller amounts but are equally critical for plant function. The macronutrients include nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur, each serving fundamental roles in plant metabolism and structure. Nitrogen is a component of amino acids, proteins, nucleic acids, and chlorophyll, making it essential for virtually all aspects of plant growth and development. Phosphorus plays crucial roles in energy metabolism as a component of ATP and other energy-rich compounds, while also serving as a structural component of nucleic acids and membrane phospholipids. Potassium serves primarily as an osmotic regulator and enzyme activator, playing crucial roles in stomatal function, protein synthesis, and the maintenance of cellular ion balance. Calcium functions both as a structural component of cell walls and as a signaling molecule that mediates plant responses to environmental stimuli. Magnesium serves as the central atom in chlorophyll molecules and as a cofactor for numerous enzymes involved in photosynthesis and respiration. Sulfur is a component of amino acids cysteine and methionine, making it essential for protein structure and function. Micronutrients include iron, manganese, zinc, copper, boron, molybdenum, chlorine, and nickel, each serving specialized functions despite being required in small quantities. Iron is essential for electron transport in photosynthesis and respiration, serving as a component of cytochromes and other electron-carrying proteins. Manganese plays crucial roles in photosystem II and serves as an enzyme cofactor in various metabolic pathways. Zinc is required for the activity of numerous enzymes and plays important roles in protein synthesis and growth regulation. The acquisition of nutrients from soil begins with the complex interactions between plant roots and the soil environment, where nutrients exist in various chemical forms and physical locations. Some nutrients are readily available in soil solution as dissolved ions, while others are bound to soil particles, incorporated into organic matter, or present in mineral forms that must be weathered before becoming available to plants. The chemistry of soil solutions is influenced by pH, organic matter content, microbial activity, and interactions among different ions that can either enhance or inhibit nutrient availability. Root morphology and architecture play crucial roles in nutrient acquisition, with plants exhibiting remarkable plasticity in root development in response to nutrient availability patterns. When nutrients are scarce, plants typically increase root growth relative to shoot growth, maximizing the root surface area available for nutrient uptake. Root hair development is particularly responsive to nutrient status, with longer and more numerous root hairs developing under nutrient-limited conditions to enhance the absorptive capacity of roots. The uptake of nutrients from soil solution occurs through specialized transport proteins in root cell membranes that can move ions against concentration gradients using energy derived from ATP hydrolysis or ion gradients established by proton pumps. These active transport systems enable plants to accumulate nutrients to concentrations far exceeding those in soil solution, sometimes achieving concentration ratios of several hundred to 1. The selectivity and regulation of these transport systems allow plants to preferentially acquire essential nutrients while excluding potentially harmful elements. Different nutrients are transported by different classes of membrane proteins, each with specific characteristics regarding substrate specificity, energy requirements, and regulatory mechanisms. High-affinity transport systems operate when nutrient concentrations are low, enabling efficient uptake even when nutrients are scarce. Low-affinity transport systems become important when nutrients are abundant, allowing rapid uptake without excessive energy expenditure. The expression and activity of these transport systems are precisely regulated in response to plant nutritional status and environmental conditions. The symbiotic relationships that many plants form with soil microorganisms represent crucial mechanisms for nutrient acquisition, particularly for nutrients that are relatively immobile in soil or present in forms that are difficult for plants to access directly. Mycorrhizal fungi form intimate associations with plant roots, extending fungal hyphae far into the soil and dramatically increasing the effective surface area for nutrient absorption. These fungi are particularly important for phosphorus uptake, as phosphorus moves very slowly through soil and often becomes depleted in the immediate vicinity of roots. Nitrogen fixation by symbiotic bacteria represents another crucial microbial partnership that enables certain plants to access atmospheric nitrogen, the most abundant gas in Earth's atmosphere, but one that is unavailable to most organisms in its gaseous form. Rhizobia bacteria form specialized nodules on the roots of leguminous plants, where they convert atmospheric nitrogen into ammonia using the enzyme nitrogenase. This process requires substantial energy input from the host plant, but provides access to an unlimited nitrogen source that can support growth even in nitrogen-poor soils. Once nutrients are absorbed by roots, they must be transported throughout the plant to meet the demands of growing and metabolically active tissues. This transport occurs primarily through the xylem, where nutrients dissolved in the transpiration stream move from roots to shoots. The concentration of nutrients in xylem sap reflects both the rate of uptake by roots and the transpiration rate, with nutrients becoming more concentrated when transpiration is reduced. The distribution of nutrients within plants involves complex regulatory mechanisms that prioritize the allocation of limited nutrients to tissues and processes that are most critical for survival and reproduction. Young, actively growing tissues typically receive priority for nutrient allocation, while older tissues may export nutrients to support new growth. During periods of nutrient stress, plants may mobilize nutrients from older leaves and transport them to younger tissues, often resulting in characteristic patterns of nutrient deficiency symptoms that appear first in older leaves. The phloem plays important roles in nutrient transport, particularly for the redistribution of mobile nutrients such as nitrogen, phosphorus, and potassium. Unlike xylem transport, which is driven by transpiration and moves primarily from roots to shoots, phloem transport is bidirectional and can move nutrients from leaves to roots, from older to younger tissues, and from vegetative tissues to reproductive structures. This flexibility in nutrient transport enables plants to optimize nutrient allocation in response to changing developmental and environmental conditions. Storage and remobilization of nutrients represent important strategies that enable plants to maintain adequate nutrition during periods when uptake is limited or when demand is high. Nitrogen and phosphorus, in particular, can be stored in various organic forms and remobilized when needed for new growth or reproduction. Vegetative storage proteins in roots and stems serve as nitrogen reserves that can be mobilized during spring growth, while phosphorus can be stored in vacuoles as polyphosphate or in organic forms that can be broken down when needed. Nutrient deficiency symptoms provide important diagnostic tools for understanding plant nutritional status and identifying limiting nutrients. Different nutrients produce characteristic deficiency symptoms that reflect their specific roles in plant metabolism and their mobility within the plant. Nitrogen deficiency typically produces yellowing of older leaves as chlorophyll breaks down and nitrogen is remobilized to younger tissues. Phosphorus deficiency often results in purple or reddish coloration due to the accumulation of anthocyanin pigments, while potassium deficiency causes browning of leaf margins and increased susceptibility to disease. The regulation of nutrient uptake and utilization involves complex signaling networks that integrate information about plant nutritional status, environmental conditions, and developmental stage. Plants possess sophisticated sensing mechanisms that can detect changes in nutrient availability and adjust their physiology accordingly. These regulatory systems operate at multiple levels, from the expression of transport genes to the modification of root architecture and the allocation of photosynthetic products. Nutrient use efficiency represents an important concept in plant nutrition, describing how effectively plants can utilize acquired nutrients for growth and reproduction. Plants in nutrient-poor environments often exhibit high nutrient use efficiency, maintaining growth rates and metabolic activity with minimal nutrient inputs. This efficiency can be achieved through reduced nutrient loss, enhanced nutrient recycling within the plant, or more efficient utilization of nutrients in metabolic processes. The interaction between different nutrients adds additional complexity to plant nutrition, as the availability and uptake of one nutrient can influence the uptake and utilization of others. Antagonistic interactions occur when high concentrations of one nutrient inhibit the uptake or utilization of another, while synergistic interactions enhance the beneficial effects of multiple nutrients. Understanding these interactions is crucial for optimizing plant nutrition in both natural and agricultural systems. Environmental factors including soil pH, temperature, moisture, and aeration significantly influence nutrient availability and uptake. Soil pH affects the solubility and chemical forms of many nutrients, with some nutrients becoming less available under alkaline conditions, while others are less available under acidic conditions. Temperature influences both the rate of nutrient uptake and the metabolic processes that utilize nutrients, while soil moisture affects nutrient mobility and root function. Modern approaches to studying plant nutrition combine traditional physiological techniques with molecular biology methods that enable detailed examination of nutrient transport mechanisms and regulatory pathways. The identification and characterization of nutrient transport genes has provided insights into the molecular basis of nutrient uptake and its regulation. These advances are informing efforts to develop crops with improved nutrient use efficiency and enhanced tolerance to nutrient stress conditions. Chapter 5: Plant Growth, Development, and Hormonal Regulation. Plant growth and development represent remarkably complex processes that transform a single fertilized cell into a mature organism capable of reproduction, involving precisely coordinated patterns of cell division, expansion, and differentiation that respond to both internal developmental programs and external environmental cues. Understanding plant development requires examining the molecular mechanisms that control gene expression, the signaling systems that coordinate development throughout the plant body, and the environmental factors that influence developmental patterns. This integration of genetics, physiology, and environmental biology reveals how plants achieve their remarkable developmental flexibility while maintaining the structural and functional integrity necessary for survival in ever-changing environments. The process of plant development begins with embryogenesis, where the fertilized egg undergoes a series of precisely controlled cell divisions that establish the basic body plan of the plant. Unlike animal development, which typically involves extensive cell migration and complex morphogenic movements, plant development relies primarily on controlled patterns of cell division and expansion within the constraints imposed by rigid cell walls. The plant embryo establishes fundamental polarities and tissue patterns that persist throughout the life of the plant, including the shoot-root axis and the development of distinct tissue types, such as epidermis, ground tissue, and vascular tissue. Meristematic tissues serve as the growth centers of plants, containing populations of undifferentiated cells that retain the capacity for continued division and give rise to all other plant tissues. The shoot-apical meristem produces all above-ground plant parts, including stems, leaves, and flowers, while the root-apical meristem generates the root system. These primary meristems maintain their activity throughout the life of the plant, enabling the indeterminate growth pattern that allows plants to continue growing and adding new organs in response to environmental conditions and resource availability. Secondary meristems, including the vascular cambium and cork cambium, enable plants to increase in girth and develop woody tissues that provide structural support for large plants. The vascular cambium produces secondary xylem, wood, toward the inside and secondary phloem toward the outside, while the cork cambium produces the protective cork layer that replaces the epidermis in woody plants. The activity of these secondary meristems is precisely regulated to coordinate radial growth with the plant's overall development and environmental conditions. Cell expansion represents a crucial component of plant growth, often contributing more to overall size increase than cell division itself. Plant cell expansion is driven by turgor pressure generated by water uptake, but the direction and extent of expansion are controlled by the mechanical properties of cell walls and the deposition of new cell wall material. The orientation of cellulose fibers in cell walls determines the direction of cell expansion, with cells typically expanding perpendicular to the orientation of cellulose microfibrils. The regulation of cell wall loosening and stiffening enables plants to control the timing and extent of cell expansion in different tissues and developmental stages. Enzymes such as expansins, cellulases, and pectinases can loosen cell wall structure to permit expansion, while the deposition of lignin and other strengthening compounds can terminate expansion and provide structural rigidity. The precise control of these processes allows plants to coordinate cell expansion with developmental programs and environmental responses. Plant hormones serve as the primary signaling molecules that coordinate growth and development throughout the plant body, functioning as chemical messengers that can influence gene expression, enzyme activity, and cellular processes in target tissues. These hormones operate through complex networks of synthesis, transport, perception, and response mechanisms that enable plants to integrate information from multiple sources and coordinate appropriate developmental and physiological responses. Auxin represents perhaps the most important plant hormone for coordinating development, playing crucial roles in embryogenesis, root development, vascular differentiation, and responses to environmental stimuli such as light and gravity. Auxin is synthesized primarily in young leaves and developing seeds, from where it is transported through specialized polar transport systems that can move auxin from cell to cell in specific directions. This polar auxin transport creates concentration gradients that provide positional information for developmental processes and coordinate growth throughout the plant. The mechanism of auxin action involves binding to receptor proteins that trigger changes in gene expression, leading to altered patterns of cell division, expansion, and differentiation. Auxin-responsive genes include many that are involved in cell wall modification, indicating direct connections between auxin signaling and the cellular processes that control growth. The sensitivity of different tissues to auxin varies depending on the concentration of auxin and the presence of other hormones, creating complex dose-response relationships that enable precise developmental control. Gibberellins represent another major class of plant hormones that promote cell elongation, stem growth, and various developmental processes, including seed germination, flowering, and fruit development. Gibberellins are particularly important for promoting internode elongation in stems, with deficiencies leading to dwarf growth habits and excesses causing excessive elongation. The biosynthesis and metabolism of gibberellins are precisely regulated in response to environmental conditions and developmental stage, enabling plants to adjust their growth patterns to prevailing conditions. Cytokinins promote cell division and delay senescence, playing important roles in maintaining meristematic activity and coordinating the balance between shoot and root growth. Cytokinin synthesis occurs primarily in roots, from where these hormones are transported to shoots through the xylem. The ratio of auxin to cytokinin influences many developmental processes, with high auxin to cytokinin ratios promoting root development, while high cytokinin to auxin ratios promote shoot development. Abscisic acid functions primarily as a stress hormone that enables plants to respond to adverse environmental conditions, such as drought, salinity, and cold temperatures. This hormone promotes stomatal closure to conserve water, inhibits growth processes that would consume resources during stress, and induces the synthesis of protective compounds that enhance stress tolerance. Abscisic acid also plays important roles in seed dormancy and germination, ensuring that seeds germinate only under favorable conditions. Ethylene represents a unique gaseous hormone that regulates various aspects of plant development and stress responses, including fruit ripening, flower and leaf abscission, and responses to mechanical stress. Ethylene synthesis increases dramatically in response to stress conditions and can coordinate plant responses to wounding, pathogen attack, and other challenges. The ability of ethylene to diffuse through air enables it to coordinate responses among different parts of the plant and even between different plants. The perception and transduction of hormonal signals involve complex molecular mechanisms that can amplify and integrate signals from multiple hormones. Hormone receptors typically bind specific hormones and undergo conformational changes that trigger signaling cascades involving protein phosphorylation, changes in gene expression, and modifications of cellular processes. These signaling pathways often exhibit crosstalk, where signals from one hormone can influence the perception or response to other hormones. Environmental factors play crucial roles in plant development, with light, temperature, gravity, and mechanical stimuli all influencing developmental patterns through specific sensory and signaling mechanisms. Photomorphogenesis describes the light-controlled aspects of plant development, including seed germination, seedling establishment, stem elongation, leaf expansion, and flowering. Plants possess multiple photoreceptor systems that can detect different qualities of light, including red, far red, blue, and UV radiation. Phytochromes represent the primary photoreceptors for red and far red light, existing in two interconvertible forms that enable plants to detect changes in light quality and quantity. The red to far red light ratio provides information about shading by other plants, while the total amount of phytochrome in the active form indicates overall light availability. These signals influence numerous developmental processes, including stem elongation, leaf expansion, and the timing of flowering. Gravitropism describes the growth responses of plants to gravity, with shoots typically growing away from the gravitational vector, negative gravitropism, while roots grow toward it, positive gravitropism. The perception of gravity involves specialized cells called statocytes that contain dense starch-filled organelles called statoliths. When the orientation of the plant changes, these statoliths settle to the bottom of the cells, triggering signaling cascades that redirect growth. Thigmotropism and thigmomorphogenesis describe plant responses to mechanical stimuli such as touch, wind, and physical contact with objects. These responses can involve rapid changes in growth direction, as seen in climbing plants that wrap around supports, or longer-term changes in plant structure such as the development of thicker, stronger stems in response to wind stress. Mechanical stimuli are perceived through mechanosensitive ion channels and other sensory mechanisms that can trigger rapid changes in cellular processes. Temperature influences plant development through effects on enzyme activity, membrane properties, and gene expression patterns. Vernalization describes the requirement of many temperate plants for exposure to cold temperatures before they can flower, ensuring that reproduction occurs during favorable seasons. Heat stress can disrupt normal developmental processes and trigger protective responses, including the synthesis of heat shock proteins and changes in metabolism. The timing of developmental transitions, such as germination, flowering, and senescence, is precisely regulated by complex interactions between internal developmental programs and environmental cues. Photoperiodism describes the regulation of flowering by day length, with different species exhibiting requirements for long days, short days, or day-neutral conditions. The molecular mechanisms underlying photoperiodic responses involve circadian clock genes that interact with photoreceptor systems to measure day length accurately. Modern molecular biology techniques have revolutionized our understanding of plant development by enabling detailed studies of gene expression patterns, protein functions, and regulatory networks. The identification of developmental mutants and the characterization of their molecular defects have revealed many of the key genes and pathways that control plant development. These advances are providing new insights into the evolution of developmental mechanisms and informing efforts to improve crop plants through breeding and genetic engineering. The integration of developmental biology with ecology and evolution is revealing how developmental mechanisms enable plants to adapt to different environments, and how evolutionary changes in development contribute to plant diversification. Understanding plant development is also crucial for addressing practical challenges in agriculture and forestry, including the development of crops with improved growth characteristics, stress tolerance, and yield potential.